Immuno-Oncology is a study of how to mobilize our immune system to fight cancer. Very important components of the immune system are our immune cells. They are responsible for fighting diseases, but also for regulating itself so that they don't fight ourselves. It is a very dynamic system constantly regulating itself. And immuno-oncology tries to understand those processes between immune cells so then therapies can be designed that influence the immune system in the right way, in the way that they fight the disease. And immune cells can be visualized in tissue, which is fantastic because if we can visualize it in tissue, then we can do image analysis and quantify them. How can we visualize those cells? We can see them on H&E, but there we cannot distinguish different populations. And there are so many different populations. Let me just show you a couple of them. Natural killer cells, T cells, B cells, monocytes, macrophages, dendritic cells, neutrophils, and many, many others. And we can visualize those different populations with immunohistochemistry markers or immunofluorescent markers. Basically, those markers that make them pop out in the tissue. They can be chromogenic markers that you can visualize in bright microscopy or immunofluorescent markers. But here are examples of markers of natural killer cells, for example, CD3, CD56, CD94, and KP46. For T cells, we will have CD3, B cells, CD19, CD20 as well. Monocytes, CD14, macrophage, CD11, CD68, CD163, and so on. Currently, this discipline is so strong that for every immune cell out there that has been discovered, there is an IHC marker. And image analysis is now a standard in quantifying them. What do I mean by quantifying? What questions are going to we ask ourselves in image analysis of immune cells? Very simple. Question number 1, what immune cell is that? And question number 2, where is it? We're talking about immune oncology, so by definition, we're going to be quantifying those cells in the tumor and around it. So the question, what immune cell, is going to be answered by the immunohistochemistry marker that we're using. And the question where, there are a couple of options. We can just quantify them in the tumor in general, and I'm going to show you an example in a minute. We might need to be more granular. We might want to know whether they're in the tumor stroma or in the tumor epithelium. In general, the concept is if they're going into epithelium, they're fighting the cancer epithelial cells, which is good. We want that. If they're in the stroma, that's already a good sign because they went into the tumor. Also good. But epithelium, even better. But sometimes they aggregate on the periphery of tumor, also known as invasive margin, when the tumor is invading non-neoplastic tissue. This is good. They're making their way to the right place, but we would want to see them in the tumor epithelium. So let me show you those regions in the tumor. High-level overview, everything that's surrounded with this purple line is our tumor. Tumor has intratumoral stroma. You can see those light pink strings in between the darker pink. The darker ones will be tumor epithelium. We have the border of the purple line with the rest of the tissue. This is the non-neoplastic tissue, and exactly where the tumor meets the non-neoplastic tissue, we have our invasive borderline. The invasive borderline is exactly the line where it meets it, and we have the invasive front, or also called invasive margin. And most of the time, we want to quantify those cells there. Why do we want to do it? Because we want to know how many of each type are located where. And when we know the type and the location, we know how to influence them to make them work better, to stimulate the cells that are killing the tumor, to slow down the cells that are regulating, that are not letting the killer cells, those who are attacking the tumor, attack it. So the moment we know which types, how many, and where, we're able to target it with a potential therapy. This immune cell component is, of course, just one component of immune oncology. There's whole tumor biology, the behavior of the tumor, and there's always this play that the tumor wants to escape, immune cells need to attack and need to free the body from the tumor. But immune cell quantification is and always will be part of the immune oncology research, which is good news because if you're an image analysis scientist, you can basically contribute to fighting cancer with this analysis. So let's look at the paper where immune cell quantification was part of the bigger research. So here we are in Nature Cancer. And this paper, Low Neoantigen Expression and Poor T-Cell Priming Underline Early Immune Escape in Colorectal Cancer, authored by Peter, Nathan, Jason, Zachary, Olivia, Haley, Alex, Daniel, Coralie, Mary, JJ, Ryan, George, Daryl, Omer, and Taylor, is discussing what helps the tumor escape, immune escape, the colorectal cancer in this case. So immune evasion, the fact that tumor wants to escape our immune system, is a hallmark of cancer. This is what cancer does. And there are therapies that want to restore this immune surveillance. So they want to activate immune cells to a higher level so that they can destroy the tumor. And those have been proven highly effective in cancer with high tumor mutational burden. So tumor escapes by mutating, by genetic mutations. And if the mutation burden is high, it means that the tumor cells have a lot of non-self antigens sticking out of the cells, so-called neoantigens. Those neoantigens can be recognized by immune cells and they will be attacked, but they do not always express those neoantigens. Here, the authors showed that patients with microsatellite stable colorectal cancer express those neoantigens, even though they have lower tumor mutational burden, but they're expressed at lower levels compared to those in colorectal cancer with microsatellite instability. So what they did here to investigate it, they modulated neoantigen expression in colorectal cancer organoids. So these are cell cultures that you can later transplant into a living organism and let them grow as tumors and investigate them. And they transplanted them into the distal colon of a mice. So pretty complex experiment, but let us go into the image analysis part of this paper. So the authors used Aiforia cloud-based image analysis platform to calculate immune infiltration. So they designed CNNs, convolutional neural networks, and trained them for 3 different cell types. And here we had a chromogenic stain. So all of those cells had a different color. Fantastic for image analysis. Black were CD8 positive cells, green were CD4 positive cells, and green and red were T regulatory cells. So in figure 2, you can see how it looks. This is tissue. In H, we see the section of the organoid. And because these were organoids, we didn't have to distinguish where exactly in stroma or in epithelium these cells were. They were injected into the mouse, and the authors wanted to quantify how many cells are in this very organoid. So we have red, green, and black stain, and they correspond to CD8 is the black stain, CD4 green stain, and red FOXP3. If we have a combination of red and green, then we have regulatory cells that are positive for those 2 markers, CD4 and FOXP3. So from the image analysis perspective, what they did, they detected all the red stain, all the black stain, and all the green stain, and then they counted objects within. And the dots here are our objects. And the objects correspond to the cells. So as you can see from this Nature Cancer publication, image analysis of immune cells is an integral part of cancer research. It's not going away. When I started in 2016, it was a huge part of pharma research, and it just grows. So get yourself a good image analysis software, for example, Aiforia, and you can help fight cancer. So you've seen in this paper, first the stain was detected, and then there were annotations made. If you would like to learn about different types of annotations and different annotations tips and tricks, there's a webinar I gave together with an Aiforia scientist. So you can click the link below, register for the webinar, and in less than an hour, learn a very cool annotation tips and tricks that are going to help you in your image analysis work for the future. And I talk to you in the next episode.